Good morning, students. Welcome to Bindu Dhadwal Chemistry Lessons. Our topic for today is nucleic acids. There are two types of nucleic acids. DNA, the full name is deoxyribose nucleic acid, and RNA. The name is ribonucleic acid. A nucleotide consists of three chemical components: a nitrogen containing heterocyclic base, a 5-carbon sugar molecule, and a phosphate group. So let us study each of them in detail. A nitrogen containing heterocyclic base. Nitrogen bases can be of two types: one, purines and pyrimidines. Purines have a heterocyclic 6-membered ring fused with a heterocyclic 5-membered ring. There are two purines, adenine and guanine. And pyrimidines are 6-membered heterocyclic rings containing 2 nitrogen atoms. There are 3 substituted pyrimidines: thymine, uracil, and cytosine. So, in purines, we have two types: adenine and guanine. These are substituted 6-membered ring fused with 5-membered ring. In case of adenine, the NH2 is substituted, whereas in case of guanine, a ketonic group along with NH2 is present. In case of pyrimidines, in this heterocyclic 6-membered ring containing 2 nitrogen atoms, if there is an amino group along with a ketonic group, it is called cytosine. If there are 2 ketonic groups, it's uracil. And if along with 2 ketonic groups, just like uracil, there is a methyl group also, then it is thymine. The purines, adenine and guanine, and substituted pyrimidine, cytosine. These are present in both DNA and RNA. When thymine is present in DNA, uracil will be present in RNA. So these are nitrogenous bases. Next is sugars. So the sugar that is used is 5-carbon sugar. In case of RNA, the sugar is ribose sugar, whereas in DNA it is beta D2-deoxyribose. So this is the ribose, beta D ribose sugar present in RNA. So you have at the second position, you have an OH group attached. When in DNA, the oxygen is missing from the OH of second carbon of ribose, so it is called beta D2-deoxyribose. Third is phosphate group. So these are responsible for linkage in the nucleic acid polymers. Phosphate group will be having PO4 unit with 3 negative charges. The phosphate group is bonded to hydroxyl group of sugar. Nucleosides and nucleotides. The molecules in which one of the nitrogenous base is bonded to a sugar molecule is called nucleoside. The nucleosides of RNA and DNA differ from each other in 2 respects. The sugar in RNA is ribose, whereas in DNA it is 2-deoxyribose. Secondly, the nucleoside of RNA will contain uracil base instead of thymine present in DNA. In all other respects, the nucleosides of RNA and DNA are similar. So this is a sugar molecule, ribose, and a base, nitrogenous base, is attached to the first carbon of this ribose sugar. Then base plus sugar will form nucleoside. This is for RNA. In case of DNA, the oxygen will not be present in ribose. This bond between the sugar molecule and base is N-glycosidic bond. Similarly, in DNA, this second carbon will not have any oxygen. Both hydrogens are present here. This is nucleoside for DNA. This is nucleoside for RNA. It may be noted that this numbering is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime in sugar molecule. This is done to differentiate it from the numbering of the nitrogenous base, where we number it with 1, 2, 3, 4, 5. In sugar molecule, we number it as 1 dash or 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. The nucleosides are named depending upon the base present. If the base is adenine, when it combines with sugar, the nucleoside that is formed will be called adenosine. Guanine plus sugar will form guanosine. Cytosine plus sugar will form cytidine nucleoside. Thymine plus sugar will form thymidine. And uracil plus sugar will form uridine. For example, here we have cytosine plus sugar combined through first carbon. This is 1 dash. So this will be called now cytidine nucleoside. When the phosphate group is attached to nucleoside, it will be attached on the 5th carbon atom here. The OH of 5th carbon will get attached with the phosphate. And we get a nucleotide. Nucleoside will combine with phosphate to form nucleotide. So when phosphate group is attached to nucleoside, the compound formed is called nucleotide. In other words, nucleotide is a phosphate ester of a nucleoside and consists of a purine or a pyrimidine base, a sugar molecule and phosphate, 1 or more phosphates. So base plus sugar plus phosphate is nucleotide. Thus, we have this base attached to sugar through carbon 1 and phosphate through carbon 5, and it becomes a nucleotide. Adenosine monophosphate. The nucleotides are abbreviated by 3 capital letters. So, for example, AMP means the nitrogenous base is adenosine monophosphate. This is deoxyadenosine monophosphate. Then this is adenosine triphosphate. This is uridine diphosphate. So nucleotides are abbreviated by 3 capital letters. And if we put a small d in front, then it is a deoxy series. That means it's a part of DNA. Nucleotides are joined together by phosphodiester bonds. Linkages, 5 phosphodiester linkages between 5 prime of sugar molecule and 3 prime of sugar molecule. So this is one nucleotide, base, sugar, phosphate. This is another nucleotide, base, sugar, phosphate. The upper nucleotide will attach to lower nucleotide through the OH present on the third of the upper one. So this will combine here to form this diester, phosphodiester linkage. 2 nucleotides are joined together by 2 phosphate linkages. So this is a dinucleotide shown here. This may be simplified. The 5 end of sugar molecule then attaches to phosphate. The phosphate is further attached to sugar molecule. 5 end is attached to phosphate. The sugar molecule's third end is further attached to phosphate. The phosphate is further attached to 5 end of another sugar molecule. On the side, it is attached to a base. This will again get attached to another sugar phosphate molecule. So this is a dinucleotide. The free end is 3 prime, which is free here, and 5 prime is attached here. So 5 end, we write from 5 end to 3 end. So 5 end attached through phosphate to sugar, attached through phosphate to another sugar, and the sugar is further attached to base at carbon number 1. Therefore, the consecutive joining of sugar unit of one nucleotide to phosphate group of next nucleotide results in a long chain polymer called nucleic acid. We can also call the polymer as polynucleotide. A nucleic acid chain is commonly abbreviated by one letter code with the 5' end of chain written on the left side. For example, if there's a tetranucleotide, there are 4 nucleotides joined together having adenosine, cytosine, guanine and thymine. Then 5 end, we will write them from 5 end to 3 end. So 5 end there was adenine, so we write A, C, G, T. 5 end is adenine, then next is cytosine, then guanine, then thymine. The backbone of nucleic acid will contain alternating sugar and phosphate bonds. This is the backbone. So this is sugar molecule, then phosphate, then sugar, then phosphate, then sugar, then phosphate, so on. So the nitrogenous base adenine is attached to this sugar molecule through its carbon atom number 1. At its carbon atom 3, it is attached to a phosphate, and at 5, it is again attached to phosphate. So sugar, this is nitrogenous base, then sugar, then phosphate, then the 5th of sugar, then 3rd of sugar to phosphate, and 1st of sugar to cytosine with nitrogenous base. Similarly, next nitrogenous base will be attached at the 1st carbon of next sugar molecule, then phosphate, then next sugar molecule, thymine is attached. So this is the backbone. It contains only of sugar and phosphate, and each nitrogenous base is attached only to the sugar molecule. Thus, the nucleic acid backbone consists of alternate sugar phosphate residues. One of the 4 nitrogenous base residues is attached to each sugar molecule. A to this, C to this, G to this, T to this. The nucleic acid backbone is as shown. This is the backbone. Sugar, phosphate, sugar. And on each sugar there is a base attached. This is how. This is one nucleotide, another nucleotide, and combining we get a polynucleotide. So summary of nucleic acids, we have a sugar, then a base, and this is a nucleoside. When it is attached to phosphate, it becomes nucleotide. If the base is adenine, the nucleoside is adenosine, cytosine, cytidine, guanidine, and uracil, uridine. This is in RNA. In case of DNA, we do not have uracil, we have thymine. And in case of DNA, we have the sugar is deoxyribose, not ribose. This is the difference. Structure of DNA. So, the primary structure, we will understand the structure of DNA with the help of this model over here. So, it is a right-handed helical structure. This is a right-handed helical structure. It's shifted like this. It's like this. This is a right helical. So, sequence of nucleosides in the chain of nucleic acid is called its primary structure. So, we have thymine, then we have cytosine, then guanine, and adenine. Then again cytosine. Then red colored is, orange colored is thymine. Then adenine. Then cytosine. Red colored is cytosine, and blue colored is guanine, and this one green colored is adenine. So this is the sequence. So it is the primary structure of the DNA. It was found by E. Chergov that the base composition in DNA varied from one species to other species. However, in all cases, the amount of adenine was equal to that of thymine, and amount of cytosine was equal to that of guanine. C is equal to G. This rule is known as Chergov rule. In other words, the total amount of purines, adenine and guanine is equal to that of pyrimidines, that is thymine plus cytosine. But the ratio AT upon CG varied considerably between species. For example, in humans, the AT to CG ratio is 1.52, whereas in Escherichia coli it's 0.93. The three-dimensional structure of DNA was elucidated by James Watson and Francis Crick in 1953 on the basis of X-ray diffraction studies. Watson and Crick proposed that DNA polymers have a double helical structure where we have two strands which are combined to each other. The backbone of this strand consists of sugar, phosphate linkages, and the nitrogenous base is attached to the sugar molecule. So each nitrogenous base, for example, adenine, will be attached to thymine, whereas cytosine will be attached to guanine. So this is by double hydrogen bonds between adenine and thymine, between cytosine and guanine by triple hydrogen bonds. So the double helical structure of DNA consists of two right-handed helical polynucleotide chains coiled around the same central axis. The two strands are anti-parallel. That is, this is the 5' end, and the other end of this strand will be 3' end. So the two strands are anti-parallel. If this is 5 to 3, then this is 3 to 5. The two strands are anti-parallel. That is, their 5'3" phosphodiester linkage runs in opposite direction. If this is 5, the other one is 3 side. So these two strands are marked 5' and 3', which indicate that the free hydroxyl groups of terminal deoxyribose units are present at 5' and 3' respectively. The linear polymer has a free 5 hydroxyl group at one end and free 3 hydroxyl group at the other end. One end is 5 hydroxyl group and at the other end it's 3 hydroxyl group. The nucleotides making up each strand of DNA are connected by phosphate ester bonds. This forms the backbone of each DNA strand from which the bases extend. So this is the backbone. This is the backbone. Sugar, phosphate, and from sugar, these are the nitrogenous bases that are extending. These are running, and so the two backbones, these are on the outer side, and on the inner side they are stacked with these nitrogenous bases. Inner side they are stacked with these nitrogenous bases. The two nitrogenous bases are connected by hydrogen bonds with each other. Adenine and thymine by double hydrogen bonds, and guanine and cytosine with 3 hydrogen bonds. So the nitrogenous bases, they are flat plates which are attached on the railing of a spiral staircase. So this is sugar, this is ester, and the nitrogenous bases form the stairs, and these form the railing of the double helical structure. So the nucleotides make up each strand of DNA, are connected by phosphate ester bonds. They form the backbone of each DNA strand from which the bases extend. The bases are stacked inside the helix in planes perpendicular to the helical axis. It's like a stack of flat plates held together by 2 ropes. These are the 2 ropes. These are the 2 ropes held by 2 ropes of sugar phosphate polymeric backbone running outside of the stack. The bases project towards each other within the structure, and the sugar and phosphate components form the structural framework on the outside. So this is sugar, this is phosphate, this is forming the structural framework on the outside. Inner side, the flat plates are present which are forming the stairs. So the bases are thus the steps of a spiral staircase, and sugar phosphate framework is the railing. Such a structure is called double helix structure. The order in which the bases occur is called the base sequence. As a convention, the bases are abbreviated by the first letters. Now, the secondary structure, the 2 strands are held together by hydrogen bonds. Hydrogen bonding is very specific because structures of bases permit only one mode of attachment. So thymine will always get attached to adenine, and cytosine always will get attached to guanine. There are 2 double hydrogen bonds between thymine and adenine, and there are 3 hydrogen bonds between cytosine and guanine. So if we know the sequence on one strand of the DNA, say A, G, T, C, A, G, we can automatically know the sequence of other because they are complementary in nature. A will always get attached to T, G always to C. So if this is A, this will be T, this is G, this will be C, this is T, this will be A, this is C, this will be G, A, T, G, C. So we get to know the other sequence by itself. So 2 strands of DNA are said to be complementary to each other in the sense that the sequence of bases in one strand automatically determines the other. So wherever there is adenine in one strand, a thymine will appear in front of it in the other strand. And the diameter of the double helix is 2 nanometer, and the distance between 2 helix is 3.4 nanometer, and there are about 20 nitrogenous base present between 2 repeating units of these strands. So when these 2 strands are heated, they automatically get separated from each other. This is known as melting. When they are cooled, they again hybridize. This is called annealing, and the temperature at which the 2 strands completely separate is known as melting temperature. This is specific for each specific sequence. That is all in today's video. If you like the video, please do share and subscribe, and thank you and have a nice day.